Our next speaker is Carlos Fernandez from University of Colombia.
Yes. Okay, good afternoon. I'm Carlos Hernandez. I'm a master's student in electrical engineering at Universidad Nacional de Colombia. So today I'm going to present the final results of my master thesis, a microbial fuel cell steady-state electrical model based on Geobacter sulfurreducens strains. Okay. Oh, sorry. Okay. First of all, I'm going to introduce you with the research we have made in Colombia. So, Then I'm going to present the electrical model proposed, the parameter identification and model validation, and finally some conclusions. Okay. First of all, our research started in 2009. So it was a student initiative. We wanted to combine the electricity production with wastewater treatment. So as you can see, our first prototype has a quite large distance between the electrodes. You can imagine how big was the internal resistance. Then we connect some prototypes in series and we obtained 1.5 volts. It was so exciting at this time for us. Then in 2010, we developed another prototypes, just improving the internal resistance. We use farm animal manure as a bacterial source, and we use synthetic domestic wastewater. We obtained some efficiency, CO2 efficiencies, so quite low at the beginning, but then we could improve that. With the prototype in the right, we obtained 2 milliamps, which was exciting as well for us because, well, as an electronics engineering, I know that some sensors can work in microamps, so that's quite interesting. Okay, so then we work with some activated sludge from wastewater treatment plants. We improve the CO2 efficiency, but the electric performance was not good. Then we were doing some experiments with sedimental fuel cells just to test, just to implement an energy harvesting system. And you can see a red light, so it was an LED, so it was good for us just to to look at that. But well, the question was, what would be the next step? So we wanted to understand better what happens in a microbial fuel cell, more from an electrical engineering point of view. So we wanted to do a model. So in 2013, I presented an oral presentation at the fourth International Microbial Fuel Cell Conference at Cairns. So we implemented a model on MATLAB Simulink. So each block represented a polarization, so activation, concentration, and ohmic polarization, and the internal potential was modeled as well. Well, in 2015, we decided also to do some experiments with pure culture bacteria, in this case, a native Colombian Clostridium from the Biotechnological Institute of the university I came from. So, well, I'm going to present this tomorrow in the poster session. So, well, the electrical model, this is the principal focus of this talk. So the idea is to identify some important parameters here, some important parameters like the open circuit potential. You can see the open circuit potential. The short circuit current as well, it's quite important in this case, the maximum power point. So we can identify different polarization across the entire voltage current behavior. So at low current densities, it is we have maybe read that the that the activation polarization is predominant. At intermediate current, the ohmic polarization is predominant. However, we have taken into account that this is true for high temperature fuel cells like solid oxide fuel cells. So maybe it is not a good idea to calculate the internal current, just measuring the slope at this region. And finally, the concentration over potential at high current densities and ohmic over potential as well contribute at this region. Okay, so the internal potential was modeled and it was taking into account the pH and species concentration, well, the pH in the bulk. So temperature and open circuit potential losses. So we know that the theoretical voltage is never obtained in this fuel cell. So we have maybe some reactant flows from one chamber to another. So the activation losses, in this case, I use Butler-Volmer kinetics, and I use the hyperbolic zinc solution. So assuming that the electron transfer coefficients are the same for the oxidation and reduction reaction at each electrode. So a rugosity factor was taken into account. This is just to to have into account the morphological effects of the catalyst at the cathode and the catalyst at the anode, so the biocatalyst. So then ohmic losses, in this case ionic and electronic resistance, it's important. So in this case the mixed conductivity of the biofilm, because the biofilm led to flow electrons and ions so at the same time, so it has these properties that the cathode catalyst has as well. So the electrolyte conductivities are quite important, are the more important in this case. Concentration losses, so in this case it's Nernst equation based. However, some kinetic effects are included, and the limiting current densities as well. So in this case, one of our unknown parameters is the limiting current density at the cathode, and we analytically calculate the anodic limiting current based on the maximum flux of substrate through Geobacter biofilms. So these are some as general assumptions. It will be modeled a microbial fuel cell based on conducting biofilms. The microbial biofilm is assumed as a conductive solid matrix with a no conductivity and thickness. The model is applied to Geobacter sulfurreducens strains, can be used in double chamber and single chamber microbial fuel cells, and it is assumed glucose or acetate as the principal carbon source. No species or activities were calculated for the internal potential. Oh, excuse me. The model is applied to stationary conditions, so I assume that the biofilm has reached the more measured state, so greater thickness and density. The substrate concentration is assumed constant across the biofilm. The oxygen concentration and CO2 concentration I assume to be constant as well. Another assumption is that the oxygen is reduced to water at the cathodic compartment. A constant pH at the electrode biofilm interface, and the double layer capacitance effect is omitted. So in this case, it's just a steady state model. So for another step, it could be interesting. So first of all, as you can see here, this is there are some constant voltage source and dependent voltage source that model the internal potential. So we know that the internal potential depends on temperature and concentration. Here we have first the cathode potential. So here first of all the constant voltage source is modeled by this part of the equation where we have the standard potential reduction potential of oxygen and some losses at equilibrium conditions. So the dependent voltage source is modeled by this part of the equation when we have the reaction quotient. So we have the relation between the products and reactants concentrations. So the same for the anode, in this case in the lower part of the circuit. So here we have first of all this part of the equation is modeled as a constant voltage source, and then we have here the dependent the dependent voltage source, but depends on current and and concentration and temperature, and we have as well the reaction quotient that relates the concentration of acetate, the carbon dioxide, and the pH. So we can calculate the potential. So to model the activation over potential, so we model as variable resistance. They depends on current and temperature. So these are the principal. I want to mention the equilibrium exchange current density is maybe the most important parameter here. So I use this model, the hyperbolic sine solution. So in this case, you can see here the important parameters, the roughness factor, A, and the equilibrium exchange current density at the cathode. So the roughness factor is this relation, so relates the electrochemically real catalyst area over geometric electrode area. So this applies for both electrodes and for the anode as well. So the activation over potential is obtained by adding those over potentials. So I found in literatures the interval range of, interval of these parameters, like the equilibrium exchange current density. There are not much information, but I think enough to start to implement a model. The concentration over potential were based on the Nernst model. So, however, here we have a parameter that takes into account some kinetics effects. And on the other hand, we have here the limiting current density at the cathode. The same for the anode. So for the anode, we calculate the limiting current density according to the maximum flow of the substrate through Geobacter biofilms. So the total over potential is by adding. So the limiting current density of the cathode was calculated with this expression that relates some kinetics information of bacteria. So sometimes it could be something confused, but if you see polarization curve at high current densities, this is not the short circuit current density. So the short circuit current density have into account all the polarizations. So this is the ideal maximum current that it could be obtained using these bacteria. So ohmic over potential was taking into account the flow of electrons from the biofilm through the electrode, interconnects, and cathode catalyst layer, as well the ionic resistance, the flow of ions through the biofilm, through the anolyte, the membrane, the catholyte, and the catalyst layer at the cathode. So these were modeled as constant resistors. In this case, the total internal resistance is obtained by adding the electronic and ionic resistances. So we know that it depends on the geometry of the cell. So to calculate the resistance of the biofilm, we take into account as well coverage factor, Fc. And this is the ohmic over potential. So here I simulate just assuming a biofilm conductivity of 5 millisiemens per centimeter. So we can see that at the highest over potential of the biofilm was 0.6 at 1 milliamp per square centimeter. So, but maybe it could be so low, but if we have maybe a biofilm with a lower conductivity, so this potential increase and we could obtain until 30 millivolts. So we should consider that. We always try to neglect the effect of the electron resistance, that it could be important here. So the ionic polarization, we can see that the anolyte catholyte is the most important in this case. So we obtain almost 0.2 volts at high current density. So this is the complete circuit. So the output voltage is calculated according to this equation. So, well, according to literature, I found out some important parameters. So I use them and I simulate a base model just to evaluate the performance, assuming some biofilm thickness, anolyte and catholyte conductivities, biofilm conductivities. There are more parameters that are not here because of the space, because it would be not great to see all that things. But this is the model obtained after the simulation. And we can see the behavior at low current densities, the intermediate behavior of approximately linear, and the nonlinear behavior at high current densities. Okay, but that I wanted to do was knowing how the important parameters, knowing the intervals they can vary. So the idea was to find out some experimental data on literature and try to estimate some parameters. In this case, we have 6 parameters: the equilibrium exchange current density of the cathode and at the anode, knowing the interval according to the information found in literature. So the internal resistance as well, the concentration parameters BA and BCA, and finally the limiting current density of the cathode. Okay, the validation was made with 30% of the experimental data. The algorithm used to do this parametric regression was the truth region reflective algorithm in MATLAB. However, we obtained these parameters and we obtained a pretty good determination coefficient. However, the question is this good? Maybe it could be an overfitting to study better this estimation. We implement a bootstrap Monte Carlo method. So having some data sets of polarization curves. So if we have a experimental polarization curve, from this original data set we can generate n new random data sets. In this case, I generate 10,000 of new polarization curves according to an original polarization. So then we're calculated all these parameters for all these 10,000 data sets, and the parameters were estimated taking into account the median of each data set of estimated parameters. So it is an statistical methods just to generate randomly data from an original data. So using that, these are all the 10,000 polarization curves obtained with each polarization curve with its parameters. So to estimate the final parameters, it was calculated the median of all the data sets. So the estimated parameters from Monte Carlo was those presented here. So maybe you could see that the equilibrium exchange current density is quite high, but it is 8.19 plus 10 to minus 4. But in this case, it's important to say that the roughness factor is included, so it multiplies the equilibrium exchange current density. And well, each parameter were within the interval we wanted to. This is a confidence interval of 75% where we find the parameters estimated. So we validate the model with 30% of the experimental data, and determination coefficient was calculated as well. So some conclusions. The model proposed was able to represent the steady state electrical behavior of a microbial fuel cell based on conducting biofilms. Well, it is important to use statistical methods to estimate parameters and avoid overfitting. It is really important to take into account both voltage losses at the anode and at the cathode in a microbial fuel cell. And the biofilm conductivity has a considerable effect in electrical resistance. So for a future work, it could be taken into account other losses like short current, the flow of electrons maybe through the membrane. So this lower the performance, and it's represented another over potential, as well as the flows of reactant through one compartment to another, and vice versa. Yes. Okay, some references, some acknowledgements to Professor Francisco Roman, the Electrical and Electronic Department, and Professor Nestor Algeciras at Chemical and Environmental Department. Okay, this is Bogota theory. This is Universidad Nacional de Colombia. Thanks a lot for your kind attention.
Have a question?
Congratulations for your presentation. Have you taken into account the membrane potential?
Oh yes. According to the conductivity, well, the ohmic over potential. So if we know the conductivity of the membrane, we know the thickness, we know the area, so we can calculate the resistance and the
But
ionic.
this is the resistance, but I mean, if you have a different conductivity between anodic compartment and cathodic compartment, you are going to have a donor potential or membrane potential or...
Yes, as well. For example, at the anode, we have a medium with a conductivity. So this conductivity can be between, I don't know, maybe, I remember 10 millisiemens per centimeter, something like that. I use this value in the simulations. And at the cathode as well, because to improve the conduction of the ions through the membrane and then to the cathode, it's necessary to have a good conductivity at the cathode. So I took into account the conductivity values at the cathode light. Yes.
Okay, thank you.
Welcome.
Any other questions?
Interesting talk. I've looked at other parameters like growth rate of the species and retention time in a fuel cell and how that can be modeled.
Okay, when I calculate the limiting current density of the anode, you mean?
Well, how fast the species is growing on the anode, so you can make predictions on parameters for flow rate and things like that.
Oh, well, I assume that the biofilm is in the mature state, so it is like a steady state. This is I'm assuming, so I suppose it achieves a certain thickness. In this case, I simulate 50 micrometers with a certain conductivity. But that's what. So I just take into account the kinetic parameters of bacteria to calculate the limiting current density at the anode. Thank you.
Thanks for your presentation.
Thank you.
Paul Rodenas from Vetsus. Have you tried to validate your model with impedance measurements, for instance, to see if you can see exactly the ohmic drop and the double layer capacitance and the...
So you mean with proper experiments?
Yeah.
Well, the point is that I have made some experiments with Clostridium. We don't know which is the electron transfer mechanisms that use this. It could be, it could be, the model could be have some changes to obtain some information, just to know maybe how important is activation or concentration. But well, I would need to think in those changes.
Yeah, because especially when from my experience in the impedance in bioelectrochemical systems, you at least you get really diffusion limitations. For instance, you can start seeing that in the impedance spectra.
Yes.
And then it would be nice to see it also in the
Yes.
in the model.
What I made was to use another experimental data in literature, so I couldn't present here because of the time, but there are like 2 more polarization curves, and I obtain information. This information, well, it's quite logic because the parameters were within the intervals, and well, I would need to do experiments to obtain better results. Thank you.
So let's take the speaker.
Okay, thank you.